The bottom line is it is difficult bringing drugs to market. It's 20, 30 years ago, people said too, 90% of new drugs that enter clinical trials fail. And I think that's roughly what it is now. So that's difficult. It shows you the difficulty of it. I think part of what people need to do is to close that gap between research and clinical development. I think people need to get smarter in terms of how they're doing clinical trials at all levels, be it from community hospitals to university hospitals, have people really engaged in that aspect of closing that gap that currently exists between research and clinical applications.
Welcome to the Personalized Medicine Podcast. This is the place where scientists, clinicians, and entrepreneurs discuss the progress of this rapidly developing field. I am your host, Alexander Yahensky. Let's start. 3, 2, 1, and we are live. Welcome to the next episode of the Personalized Medicine Podcast. Today, we are returning to a very exciting topic of immuno-oncology, and we will talk in detail about novel approaches to treat cancer based on CAR T-cell technology. It is my pleasure to introduce to you Dr. Eric Von Hoffer, the Senior Advisor and former President of Affyimmune Therapeutics. Eric's story is a showcase of a remarkable career in the biotech industry. Eric studied biology at UC Berkeley and received his PhD from the University of Southern California, where he worked on genotoxicity studies of cancerogenic petroleum fractions. Eric continued his career in cancer research doing 2 postdocs at University Hospital Zurich and at Harvard, and then taking a position of assistant professor at the University of Massachusetts Medical School. In 1992, Eric made a transition to biopharmaceutical industry, leading the development of antisense oligonucleotide therapies at Hybridon. Eric subsequently led multidisciplinary drug development programs at Millennium Pharmaceuticals as a director of drug discovery research. Eric has been fundamental to 2 prominent immuno-oncology companies, Antigen Express, a company focusing on a peptide-based cancer vaccines, and Affyimmune Therapeutics, a biotech startup developing novel approaches for CAR T-cell-based therapies. With about 50 articles in peer-reviewed journals and 4 patents, Eric is a true expert in the field of oncology, and it is my privilege and honor to welcome him on our show. Eric, thank you so much for accepting my invitation and welcome on our podcast. My pleasure. Good to be with you here. Perfect. So, Eric, you have a very fascinating and inspiring career and working both in academia and in industry. So, can you describe the arc of your journey for us? What got you inspired in biology in the first place, and how that interest led you through the amazing career steps that you have experienced so far? Sure. Yeah. So my interest in biology really started when I was very young. I think probably when I was in elementary school, I was just fascinated with biology. I remember the first time looking at the simple things one does in school, looking at an onion skin under a microscope, seeing the beautiful organization of cells and whatnot. And the more I looked into that, the more I could see this beautiful kind of organization that there was in biology and nature. So that got me started at an early age. I was also fortunate when I was in high school as a teenager. I had the opportunity of working at a hospital in Los Angeles under the direction of the head of surgery there. And again, that's where I really began doing cancer research. So this was really wonderful. I mean, every time when you work at school, you have a very limited kind of opportunities. Here, this was actually working on things more directly related to patient care. So that was probably one of the most motivating things in my career, and it really kind of evolved from there. I continued some work during college, and then in graduate school, it became clear to where my interest was in terms of cancer research, and it really kind of took off from there. Perfect. And speaking about cancer, I think you've been working in the field of oncology for decades now. And I'm just curious to hear from you, what is your take on the evolution of the approaches that we apply to treat cancer, both from the scientific and the medical standpoint. And what is the role of immunology in that oncological treatments?
Yeah, no, good question. There's been a lot of change over the last 30 years. I think one saw in the 1990s kind of a plateau in terms of progress one was making against cancer. Immunology, people were kind of fascinated with this. But there were, anecdotal bits of evidence for the role of the immune system in oncology. So I recall in the 1980s, people would do little experiments where they would irradiate tumor cells for murine tumors, inoculate a mouse with those and like a vaccine. And occasionally you would get regression of tumors in the mice. But people really didn't understand immunology enough to be able to apply this in any really practical way. So a lot of it was just waited for basic research to be done in the field of immunology, so that people could really get a handle on the complex immune system. Part of that too is the gap between experimental systems, model systems, and what happens in people. So animal models will give you some information, but at the end, they clearly lack. They don't predict that well what you're going to see in the clinic with any model, and I think that's particularly true in the field of immunotherapy, because mouse immune systems is different from human immune systems. So it's really a big jump. So I think a lot of it had to do with how the knowledge has evolved in the field of immunology and immunotherapy before being able to apply it. And I think that, certainly in the last decade, with the advent of, really two things. One was in the early 2000s with adoptive immunotherapy. This was work by C. Rosenberg at the NCI, showing that he could get a 50% response rate in patients with metastatic melanoma, and that those data have persisted through today. That really showed that if you had the right T cells, you could have a curative effect in cancer. And then clearly, from 2010, people there was really the advent of checkpoint inhibitors, also showing very impressive responses to many cancers, albeit it's in a roughly 30% of patients, 20 to 30%. So, but again, this was something in an area where previously there was really no. There's a lot of patients for whom there were no other types of therapy. So, two very important points. One on actively showing that active immune cells could do something. And then the other side of that is that if you take the brakes off the immune system, you can also have a very good effect.
Perfect. Yeah, and let's talk maybe a little bit about the first component and also about the Affyimmune Therapeutics. I'm curious to know what actually Affyimmune Therapeutics is doing and how the CAR T cell based therapies that you are developing are different from what other companies in this space are doing.
Sure, right. So what's different about Affyimmune is that they're using affinity-tuned CAR T cells. So what that means is that they're taking CAR T cells targeted to tumor antigens, but they're tuning the affinity so that it's optimal in terms of getting maximal activity of the T cell against the cancer. Up until now, the majority of people simply take either a single-chain monoclonal antibody, which binds the most tightly to the target. And then they use that to essentially target the CAR T cell. But these are not really physiological levels. So generally this is CAR T cells are targeted with single-chain monoclonal antibodies that bind in the nanomolar KD realm, which is about 1,000-fold more tightly bound than normal T cell receptors. Normal T cell receptors identify their targets and bind in the low micromolar range. So what really appealed to me is that what you're doing in terms of affinity tuning is you're making it more physiological relevant, and you're preserving more of the natural T cell function. So as I indicated, people have seen that if you get the right T cells, those cells on their own can kill cancer cells. And I think by affinity tuning, you're basically preserving those functions. So you get less T cell exhaustion, you get better serial killing, so going from one tumor cell to the next. And the other advantage there as well is that most tumor antigens are not really tumor specific. So in hematological malignancies, it's somewhat of a more rare issue where you have CD19 that's specifically expressed on tumor cells, also B cells, but B cell aplasia one can deal with clinically. Most cancer cells are truly just associated. So you have some low level of expression on normal cells. So again, if you have affinity tuned CAR T cells, you can tune them such that they will not kill normal cells with low basal levels of expression of a target, but they will very effectively kill tumor cells that have higher levels of expression. So it's really two things that the company is accomplishing. One is to preserve better T cell function, get more longer lived T cells, and also to avoid what's called on-target off-tumor toxicity. So you're making it safer for the patients as well.
So sounds fantastic. So I'm curious about the technical details of this affinity tuning approach. So how that can be achieved practically?
Right, excellent question. So the CAR T cells, again, the affinity tuned, what we've done is looked at the that which is used to target the CAR T cells, so either single-chain monoclonal antibodies, and simply by various methods, modifying those single-chain monoclonal antibodies or natural ligand by mutagenesis or some other means, such that they do not bind quite as tightly. And one needs to do a number of steps to ensure that one still is getting specificity and yet lowering the binding affinity. So it's really molecular biology, mutagenesis approaches. Actually, it's an interesting story for the founding of Affyimmune. One of the initial targets we're going after, ICAM1. Studies were done years ago by one of the co-founders of Affyimmune, Moon-Soo Jin, in the laboratory of Tim Springer at Harvard, where he was looking at the interaction between the target, ICAM1, and its natural ligand, LFA1, lymphocyte function-associated antigen 1. What's interesting with that interaction is the LFA1 can actually change its conformation in a way that changes the affinity. So what Moon-Soo Jin was doing, who's now a professor at Cornell Medical Center, was to do directed mutagenesis by a variety of means to get a series of different affinity mutants that bind to down to ICAM1 all the way low nanomolar up to the micromolar level. So at that point, it was possible to do a structure-activity relationship, which one does all the time in small molecule drug discovery, but has not been done in CAR T cells up until now, really. So that was the perfect opportunity. Once he had these different affinity mutants, simply put those onto CAR T cells and then look at a variety of studies, both in the laboratory and in animal models, to identify which is really the optimal CAR T cell.
Perfect. And I think it's a great example, again, of how fundamental science is important to drive our progress in the world of biopharmaceuticals.
Very true, yes.
Perfect, yeah. And I'm still constantly impressed by the power and importance of those cells adhesion molecules, right? And what you just mentioned is super fascinating, right? If you can just tweak a little bit the structure of those ICAM molecules or perhaps other cell adhesion molecules, we can change the interaction between the cells and hence fine-tune therapeutics. Sounds really exciting. We are doing this show for you, and your feedback is very important for us. So if you have any suggestions or comments, or would like to recommend a guest, please write us an email to team@pmedcast.com. You can also reach us on LinkedIn, Twitter, or Facebook. Just type in Personalized Medicine Podcast and you will find us there. To catch our next episode, subscribe to our podcast on your favorite podcasting platform. Please rate us there and leave a comment. This helps us make this show better and reach more people interested in personalized medicine. And now, let's get back to the interview. Eric, one more thing that got me thinking when I was looking through the pipeline of Affyimmune were the bispecific CAR T cells. So can you tell our audience what those are? And again, how easy or how difficult is it to produce them?
Right. So, one issue with any type of targeted therapy. So, anytime one identifies a target on cancer cells that you attack that target, you can kill the tumor cell. Tumor cells tend to fight back at you. So, what they do is simply, identify a way of losing that one target, and they happily then grow. So, if you have multiple targets, you're cutting off an escape route of that particular tumor. So the other interesting thing about the first target we're going after, ICAM1, is that's actually inducible by a cytokine released by activated T cells. So when T cells, CAR T cells, any other type of T cell become activated and start killing tumor cells, they'll release interferon gamma, which is a cytokine. That cytokine happens to upregulate the expression of ICAM1. So you're forcing the tumor to do something it doesn't normally do, which is to increase the expression of a target rather than decrease the expression. Now, that's interesting in a dual CAR-T context, in that if you had some other you're targeting some other target, like, for instance, mesothelin or some other target, no target is expressed in 100% of the tumor cells. They're expressed in maybe 60, 70%. So if you then start killing those cells that are expressing that particular target, the T cells, the CAR T cells, will then in that process secrete interferon gamma and upregulate the expression of ICAM1 in the remainder of the tumor cells. So you're taking advantage of multiple targets in the tumor with this dual CAR T strategy. So people have looked at that a little bit in hematological malignancies, but we're pushing to develop that in the context of going after solid tumors.
Essentially, from what I understand, the rationale would be just to improve the efficiency of that CAR T cell therapy against a different subpopulation of cancer cells.
Exactly. Sure, you're killing a broader spectrum of tumor cells.
Got it. Super exciting. Perfect. Eric, I think one more topic that I want to discuss with you is related to your previous work at Antigen Express. I think the topic that is also very hot in oncology today are the cancer vaccines. And I know that at Antigen you worked on the peptide-based cancer vaccines. Now we have obviously the explosion of mRNA technology in the industry, partly driven by the COVID vaccines. In your opinion, will the application of anti-cancer vaccines, being it peptide or mRNA-based, improve over the time? And if so, which specific cancer indications do you see as the front line for the use cases for that technology?
Right, sure. Well, we've definitely seen a tremendous increase in the interest in vaccines since COVID. It's really kind of interesting to see an mRNA vaccine that has not been used up until now in any context, to see that suddenly then approved in a prophylactic setting. So that's combined with, I think, as I indicated, a lot of the increase of knowledge just in cancer immunotherapy in general is giving a big boost to the field of cancer vaccines. So as well as combinations with the checkpoint inhibitors. As I indicated, they're kind of a yin and yang a little bit. Anything one does to activate T cells, be it CAR T cells or cancer vaccines, is really kind of stepping on the gas to activate the immune system, while checkpoint inhibitors are taking off the brakes. And there's always, tumors have gotten, many cancers are very good at secreting various cytokines that suppress immune cells. So I think the combination of those two, of checkpoint inhibitors with cancer vaccines, other type of immune activating technologies, will be very beneficial. So I think there's clearly been an increase over the last few years in the number of trials going on with cancer vaccines. I think that'll increase. In terms of the types of cancer people are going after, I think it still remains, those patients that have a lower disease burden. So patients that have, not bulky disease, they've gone through initial therapies to reduce the size of the tumors, and you're really kind of trying to activate the immune system to, kind of mop up the rest, as it were. I think that's still the population that people have the most interested in regards to immunotherapy, cancer vaccines.
Got it. And in terms of approach to the vaccines, do you think that it will have to remain personalized, that we would definitely need to process the tumor material from each individual patient and then tweak that either mRNA or peptide-based vaccines to that individual patient? Or there is a chance that some off-the-shelf anti-cancer vaccines might appear in the market at some point in the future?
Right. So people began a number of years ago, maybe 5 years ago, looking at so-called neoantigens. So most of those are what are called private antigens for the individual patient. It still takes a fair bit of time, say something like 3 months, to generate a vaccine, an immunotherapeutic vaccine based on those neoantigens. And I think some of the hurdles are the same that when encountered with more off-the-shelf vaccines that people have been developing. So they're still ongoing, but I think probably, I think these the more common targets, not the neoantigens. I think they're not going away anywhere. I think that those are still good targets, and people have really kind of validated many cancer targets, not just with CAR T cells, but also with antibody drug conjugates, various monoclonal antibody therapy. So there are a number of good targets out there which are common, for which one can make them off the shelf vaccines. So I think it's going to be more a challenge of identifying the right patient population, and at what point in disease is optimal really for introducing some type of activating cancer immunotherapy.
Perfect. Understand. Great. And Eric, since we speak about the future, what I would like to know, what are the 3 things that you are looking for, you expect to happen in the field of immuno-oncology over the next, let's say, 5 to 10 years?
Sure. Yeah. Now, the first thing is translating some of the success that people have seen in hematological malignancies in patients with solid tumors, and that's clearly the focus of Affyimmune. I think they've shown really good progress in that regard. So that's the first thing I'd like to see. See CAR T cells, the activity that's been shown in hematological malignancies translated into solid tumors, which really comprise something like 90% of the cancers. That's the first thing. Secondly, I think it'd be, and I think people are clearly discovering now how to do this, is to generate a more long-lasting immune response. Many of as I mentioned, there's been great success in patients with hematological malignancies, but there are many relapses. To really get a long-term remission, to really make use of the immune system as one originally envisioned, which was to activate the immune system so that it's the way we're dealing with infectious diseases like COVID. So, okay, maybe we'll need a booster shot every couple of years. That's no big deal if you have cancer, and it keeps it at bay. So I think definitely in the next 10 years, we'll see progress in terms of managing cancer in that regard. I think the other thing is we're, there's a tremendous amount of information that we're getting, both from many different patients, many different studies, and also with individual patients. A lot of the technologies now, we're looking at, single cell sequencing to look at, what exactly is going on in that patient. So I think in the next 10 years that, things like artificial intelligence and machine learning will be applied. And that could really kind of change the way people go about, both in a research aspect and a clinical aspect in developing drugs. So I think there's going to be clearly growing pains of that in that quest. But I think definitely in the next 10 years, there's going to be pretty significant inroads in terms of applying those tools. So I think, as people said before, you don't need to worry about computers taking over your job. But I think you should worry about people that know how to use AI and machine learning taking over the jobs of the people that don't know how to use those tools. So I think in the next 10 years, that's going to be probably somewhat transformational in not just cancer immunotherapy, But I think clearly in that field as well, there are many different factors that play into it, and I think sorting that out is going to require some of these more advanced tools.
Definitely. Yeah, it sounds like a very busy but also exciting future for the next 10 years. And let's hope that all of those things will come to fruition. And speaking about bringing things to fruition, the one thing that I wanted to ask you about is your experience in bringing drugs and new therapies to the market, and your view on the process of that, and has it changed over the last 20, 30 years that you spent in this industry, and where do you expect it to evolve going forward?
Right. Yeah, no, it's the bottom line is it is difficult bringing drugs to market. It's 20, 30 years ago, people said too, 90% of new drugs that enter clinical trials fail. And I think that's roughly what it is now. So that's difficult. It shows you the difficulty of it. I think part of what people need to do is to close that gap between research and clinical development. I think that's part of it. So I think people need to get smarter in terms of how they're doing clinical trials, and really at all levels, be it from community hospitals to university hospitals, have people really engaged in that aspect of closing that gap that currently exists between research and clinical applications. So, as I mentioned, animal models always have their limitations. I think we're getting to the point now where hopefully we can get information from early stage clinical trials that would give us a better information. Everything from which are the optimal patients that we can enroll, that we would expect to see a response in, to the design of novel treatment strategies. So I think it's still difficult. It's changing in that regard. I think other things too is it's clearly a lot of the fields are more crowded than they used to be. I know 5 years ago, I think there was almost nothing for, say, triple negative breast cancer. So if you wanted to do a trial in triple negative breast cancer, it was relatively easy. Now they've made inroads, which is a great thing for patients, but it's an added hurdle for identifying your that patient population that would be most likely to enroll in a new study. So I think that's maybe been added on to it.
Got it. Yeah, there is certainly no shortage of challenges in this space. And going back to your point, bridging this gap between science and then pharmaceutical industry, I'd like to ask the question that probably a lot of the younger listeners in our audience are wondering about. What type of advice would you give to the people who are thinking about transitioning from academia to industry, and either by joining an established pharma company or starting something on their own?
Yeah, no, that's always a big decision. I love that part of biotech where there's really good science, solid science coming from academia, and is really ready for more resources. However successful it was in academia, it's always a little bit of a microcosm. You just can't get the funding from grants to really do good sized clinical trials. So that's kind of the excitement of it. That I think if people have that sense of excitement about really bringing something forward, definitely they should follow it. I think part of it too is just from a practical perspective. It's good for young people considering it to try and get a little bit of a sense of the business world, the financial world that has really evolved tremendously around biotechnology. I recall when I first made the transition from academia to biotechnology, that in academia, you write papers, you get grants, and you have a pretty good sense of what's keeping the ship afloat, right? But when you get into biotech, it's different. It's different, and sometimes you just really don't have an idea exactly what's keeping it up. So I think it's important for young people to look from that perspective, to get a sense of how that financing in biotech works. And there are many biotech companies around where I think it's not that difficult to get a sense of that. And clearly, one needs to be cautious as well. I mean, a lot of the funding comes from Wall Street, in addition to other, lots of investors there. But Wall Street's like a big city. There are good parts of town and not so good parts of town. So one needs to be a little wary of that. And the same thing with investors. There's some great investors out there. There's some other investors that they're looking to make a fast buck, and that's kind of it. So I think it's important for young people to try and definitely shouldn't hesitate putting their toe in the water of seeing what it's like. But it's an exciting, very exciting career, and I can highly recommend it if you're up for the rough and tumble that is biotech.
Great, Eric. And if I may ask just one more personal question, what was the experience that struck you most after that transition to the industry from academia?
It was really the commitment of a variety of people involved with that first company. As I mentioned, it was hard to tell what's really keeping the ship afloat. But I found out later that actually in that particular company, the Hybridon, that we were close to making our first deal. And this was with Hoffmann-La Roche Pharmaceuticals, a very big deal. We had the opportunity of doing a deal with another company before that, a smaller company, for I think it was $5 million, a fair bit. But what I found out later was that the CEO and president took out a second mortgage on their private homes to make payroll because they knew that Roche agreement was coming. So seeing those types of things, seeing the commitment of people there and the passion they have for it, that's also inspiring. So that was one of my first kind of initiations into the biotech world.
It does sound inspiring, and it should be so. And I hope that lots of our listeners will hear this story and also realize that this passion and motivation can go a long way if it's put into the right things. And let's hope that more and more people will do so, right, and help our field, the world of biotech, grow further, building upon great scientific ideas.
Right. No, yep, absolutely.
Great, Eric. That was my pleasure to have this conversation with you. You are such a role model in the biotech space. I really liked everything you described of the CAR T cells, cancer vaccines. It was super insightful. I'm sure our audience will learn a lot from this conversation. One last question before I let you go. Where can our audience find you online in case they would like to reach out to you?
Sure. They can email me at evhofe@gmail.com.
Perfect. Eric, thanks a lot. I wish you best of luck for your next steps, and we'll definitely stay in touch.
Likewise, good talking with you.
Thank you so much for being with us today on the Personalized Medicine podcast. If you like this show and know someone who would enjoy it too, please share this podcast with them. And don't miss the next episode yourself. Subscribe to our podcast on your favorite podcasting app. Please rate us there and leave a comment. That helps us to grow and deliver best experience to you. To access the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. Our show notes include guest bios, links to their most notable work, and recommendations for additional reads on the topic of the episode. And if you have any feedback or would like to suggest a guest, write us an email to team@pmedcast.com. or reach out on LinkedIn, Twitter, or Facebook. Thank you very much, have a great day, and until next time.